I'm going to just tell you right now, it's a freak show. What I'm going to show you is crazy. They can make blank strands of mRNA, which means they don't code for anything specific, so they think they're fine to make. And they're squirting them in to these frog cells, the eggs of frogs. And then they squirt in a whole batch of extra ones, which are not supposed to be in there. And then they ripped the thing to shreds, and every shred they ripped tried to turn itself back into a new membrane, because there is nothing that is without membranes in any creature that lives, or anything that lives. Trees and plants and bugs and you and I and everybody. Every single cell has its own membrane around it. Even the cells in the trees, everywhere. So the key here is they're making synthetic mRNA. I don't know about that. But specifically what they did here is they took all these eggs, like these eggs of frogs, and they went, I'll show you, and they shot in this blank DNA, just or mRNA, and then every one of them tried to make its own membrane, because that's the first thing everything does, is make a membrane. And I actually could see stuff that I could recognize in that membrane, and I think I could see what I call a spur lock, which means where they latch together to fuse. Because a membrane doesn't just all of a sudden pop from nowhere. It has to basically grow and then fuse together. And that's what the spur lock does. Now, this is like Frankenstein stuff. Look at this. These creatures occupy a third state beyond life and death, scientists say. This just came out. Now look at this. This is, I forget who's doing this research, but it's some university, I'm sure, getting some funding from somewhere. Hold on, let me just figure out where this is. Oh, from Tufts. Tufts is a pretty good school. My father went to Tufts for metallurgy and did extremely well, as he told me many times. Now, and he was a very smart guy. Now, here's what they really did. They make it sound like they could just transform cells. No, these were skin cells extracted from dead frog embryos. So the frogs were developing, and they died. Well, as far as I'm concerned, are you alive or are you dead when you're developing? It's starting to look like you may not be completely alive. I don't know how, like they say, these are on the margin of life. The researchers found that skin cells extracted from dead frog embryos were able to adapt to new conditions in the petri dish. In other words, they still continued to grow, but they didn't grow into a regular embryo because they just tore them to shreds. They tried to create something, and they turned into little balls. Now let me show you the video. Again, this is Fair Use Act. I'm not trying to steal anything from Tufts. And my father went there on a GI Bill after he defended our country during World War II. He was a bomber pilot. Now, so Tufts, please don't block me. Now, this is their video showing what happens to these frog embryos. So this is an embryo. And what does that mean? It's going to grow into a frog. Inside of there is all kind of genetic material that's hooking up to start to grow into a frog. Now, at this point, is that a living thing or is it not a living thing? It's an embryo. It's making all kind of connections. But when they tore it apart into shreds, it's continued to make, well, I'll show you what it did do. I'll just play it and you listen and watch. Okay, my friends, all I can say is this is disturbing. When you read the fine print about this is from Tufts. Scientists create the next generation of living robots. How did they do that? They took amphibian eggs, which are frogs, and they injected them with RNA. So they take the eggs of a frog, which is supposed to develop into a frog, and they squirted a whole bunch of RNA in there. Well, what are RNA? It's half of DNA. So it wants to start to create something, and it's going to try to sink in with this stuff, and they just destroyed this. Listen to this. This is, I'm telling you, it's disturbing. And again, I'm not trying to steal anything. This is, I have a right to show this as a fair use. Now, I have it running slow. They're going to shoot stuff into each one of these. Bam! They just shot in a blast of that micro-injected RNA. So they're shooting each one of these. Bam! Bam! A ton of little RNA strands in there. Now, how they did it, microRNA, I'm not sure how they exactly did it, but they shot them in there, they injected them. Then they cut them all open and ripped them. 24 hours later, the egg membrane, which everything has a membrane, every single thing has a membrane. That's why they're going to try to develop new membranes. Now watch what happens. 24 hours later, the egg membrane is torn off. They just shredded it and ripped it to bits. All right, so they're grabbing it and pinching it and ripping it. There they go. You got to get a good pinch on it. Once they do it, they'll tear open. Now, stem cell tissue is then harvested from the embryo. Stem cell tissue, which is basically embryonic cells, because they're going to end up creating everything. And that's all this goop. All right, so they cut it all open. They rip off the membranes, and again, every single thing there is has a membrane. Even trees and plants and everything. And then the tissue forms into a sphere because it wraps with its membrane. That's developing a new membrane. I'm going to show you something. When it locks together, it looks to me like it almost has the spur lock. I'm going to probably stop it when it comes to that point. But it develops all of these. The tissue forms into a sphere. They're all trying to make a membrane around something. That's how the egg forms. And they're confused. They have no idea what they're doing because they've been shredded. But they all have a membrane starting to grow around, or has grown around most of them. Now this is 4 days after they become spears. They become mobile. So 4 days after they're spheres, they start to get mobile. Then they swim. You see, they're pushing all these little particles away from them. So there's some kind of, I would say, sort of a magnetic interaction here where, although I don't know, but you can see they're obviously awake. All right, they start swimming. Now, each one of them is trying to find its own way in life. Swimming is propelled by small hairs beating on the surface. So they're just, basically they're swimming, but I don't know if they're really swimming. I think they're looking for something to grow into. And they're, normally that would start to develop, and the DNA that was normal, right now it has no normal DNA, I don't think. Swimming is propelled by the hair beating on the surface. Now, if they were normal cells, they just continue to use that DNA to form the body and the structure and the arms and the legs and the ears, and they just read the codes and build it. Now, then they say that individuals can move through these mazes. Well, what does that mean? Is it in a tube? I think that it's in a tube. I don't think it has anywhere else to go. So it moves around. Let's see what it does here. It comes up to, does it go this way or does it go that way? Well, it goes the way it's headed. I don't know if that's a maze. And they're talking about a little tube, but obviously it's going to go out through the tube. And then it just takes off and starts swimming away. See you later, alligator. They don't know what to do. It came out of a narrow capillary. This is, they say, recording experiences by changing color. These are different transition metals. The only way that you can get those is different enzymes. These had to be in a little bit of different conditions. And this is the green and the red. This is the blood colors. And the green, you see the blood colors in the green? And to do oxygenation, you need copper, which I believe is the green, and you need the iron. Those two are absolutely essential as you're transpiring, changing through the color scheme of blood and using the copper as an enzyme. The groups can move particles in their own environment. They're lost. Look at them. Where do I go? What do I do? Help me! Ah! I think this is what's going on at Fermi Lab. What do I do? I don't know. Just keep walking in circles. Whoop! There's a Yale. You know, this is a new photo imaging of Yale. The groups can move particles in their own environment in circles. I guess we did evolve from pond scum. Anyway, see, they're coming back together. Now, I thought I could see a spur lock somewhere. They can also self-repair when injured. They got to come back and latch to each other. And they do. They rebuild themselves. There's another one cut up here. See that? Look at that. Watch this. You see that particle? That's that little latch. I'm telling you, it almost looks exactly like it. I've shown it 1,000,000 times. My particle here has basically the same latch, my rock here. And that's how they form and they lock together. And in certain areas, there's a whole batch of these that lock together to form connections. But my, oh, where did you go, rock? I don't know. Oh, here it is. That's the latch right there. I call that the spur lock. And that takes this fascia, this coating here, and it latches it in to another place, and the other thing would have latched right in there. So you got these locks. You see it? That latch is the same as that lock right there. I think. You know, everything little is just as above, so below. And I'm telling you, that seems to be a fact. And I'm not kidding. Now it says they can repair themselves by relatching their membrane. I'm not kidding. Look at it. Everything wants to be enclosed in a membrane. That's how you protect it. You protect it. All right, Tufts. Thank you, Tufts. All right, I know I was out of focus there a little bit, but this right here is the spur lock. That little tab locks the fascia into the next thing because it's one huge sealed system. Now these are embryos. These were supposed to be eggs, and they shot a whole batch of extra mRNA in there, messenger RNA. Now how they did it, I don't know. And what it consisted of, I don't know. Was it just a backbone without, I can't, it's got to have some amino acids in there. They must have injected something in there. But when they ripped these things apart, as I believe I showed you, when they came back together, I believe that's a spur lock. That's how they latch from one to the other. And even that little thing coming down the center could be the fluids that come off of the membrane and latch into the next part of the membrane, because as you grow, a body part comes out this way. Well, it's going to have a whole different set of circumstances surrounding it. If it's an organ, it's going to have something that has to deal with the chemistry of that organ, like a kidney or a liver or a heart or lungs. They all have different chemistry, and they will all have a different membrane chemistry, I believe. And we could test this now, because right at that spur lock, as I showed in the microscope, I'm going to show you right now, there's two little tiny spots. And they are the things that empty the fluids from here into the next attached part of the body. And if those two fluid things got clogged up, you might have a problem. And is the membrane fluids up here completely different than the fluids that'll be down here? And I think they might be. They might have different enzymes living in them. I don't know how that would happen. But I will show you the little feeders here. Now, could we put something in a person's body that would be like give off a glow, basically, when it was irradiated? I'm not talking about like nuclear radiation. I'm talking about some not real serious invasive spectrum. Almost like, you know, they do all kinds of imaging now. Something that would tell you, is this section here lit up just like all the rest of the sections? In other words, is that same fluid flowing through the entire body? Or is it only restricted here and there? And is it maybe the kidney's clogged up and it can't get any fluids flowing through there? Maybe those two little pores don't work on that kidney anymore, coming right out of here. So the kidney can't get any of that fluids in. That could be. How would we tell? If we put a fluid in the body that just washed through this entire it's basically the lymphatic system. And it went through this whole fluid-filled highway. That's what they call it now. It's a gigantic system supposed to cover the entire body, and it does. And it feeds from one bag that contains whatever it contains. It's a lung, it's an organ, it's a kidney, it's a, you know, muscles, tendons. They're all in their own membrane. Every single thing, even red blood cells, have their own membrane. All right, so once that membrane protects whatever is the contents, and the chemistry changes dramatically in your body. Dramatic changes in chemistry. One area has all kinds of enzymes and bacteria and this and that. Another area is pristinely clean. They're going to have different membranes. Something's going to be different. And this has been a totally ignored subject. Nobody even knew that membranes existed that I'm talking about until I found them basically in 2015. The paper was written in 2018 by somebody in New York University about this layer that I'm talking about. But now this is the new thing that's got to be tested, which is the spur locker. And I'm going to show you right now, at the tip of this thing right here, all of the fluids that ran around this muscle congregate right to there, and they flow through the two little holes into the rest of the, wherever it attached to. I don't know, the shoulder or a bone, I don't know. I'm going to show you in the microscope right now. Remember, we're going to be looking at that spot right there. You see it? You see that spot? Now, you see how it's like feeding all of that tissue together? Everything wants to be in its own membrane. Everything is coated by a membrane. See, this is it right here. Every single cell in biology. I asked it, does every cell in all biology, that's plants, trees, fungus, everything, every cell, do they all have a membrane of phospholipids? Yes. Every cell in biology organisms has a membrane composed primarily of phospholipids. The phospholipid layer forms the fundamental structure of cell membranes. Without them, they get invaded and is destroyed. Every single one, everything, the cell are protected by that membrane. They just never understood this. And that membrane is saturated with enzymes. That's why it's two layers. It's phospholipids, it's bilipid. In between the two membranes, all this stuff lives, which it kills anything to try to come in. It's not that hard to understand. Now, the phospholipid bilayer forms the fundamental structure of cell membranes, providing a barrier, yes, that separates the interior of the cell from external environment which tries to attack it. The membrane is also embedded with proteins, cholesterol, and carbohydrates. What are proteins? Proteins are the enzymes. Where do the enzymes come from? They come from bacteria. That's the key right there, is the proteins. And then they break down into cholesterols and carbohydrates and play various roles in cell functions. They provide the energy, they provide the building blocks, they provide the immunity, they provide everything. This doesn't special various roles, it plays basically all the roles. It only allows in what it will allow in. It only allows out what it will allow out. Its organelles inside there facilitate the factory that does whatever that cell is supposed to do. Some of them make enzymes, some of them make bone, some of them do all whatever. But without the membrane, you're done. It's over. Case was closed. Now, they say the membrane is embedded with proteins, which are the enzymes, cholesterols and so forth, for the cell function, communication, and transport. While there are variations in membrane structure among different cell types, prokaryotes and eukaryotes, the presence of a phospholipid membrane is a common feature of all living cells. Phospholipids. They break down into phosphates. You see this? Phospholipids and phosphates are related. They actually break down in the cellular structure depending upon how much energy is being used out of them and how much light is hitting them and so forth. It's in ATP, it's in DNA, it's in RNA, it's nothing but life. Phospholipids, when you see that, you know you got life, and guess where that is? All right, there's layers of phosphates because the muscles are in layers. And there's a layer of phosphate, or phospholipids, then there's a layer of muscles, and then another layer. And that's what they discovered. The discovery of phosphate layers in the samples from asteroid Bennu collected by NASA is significant. It suggests that the building blocks of life, which this is what it is, phosphate surprise they called it, may be more widespread in the solar system than previously thought. Phosphates essential for various biological processes, their membranes, including formation DNA and RNA. It's biology. And now they're going to study this for years and so forth. Well, let me show you what it looks like. It is muscle sarcomeres. We don't have to study things for a bazillion years. We need to discuss what its impact on society is and our meaning of history and so forth. That's what we need to start talking about. This has all been done. It's all figured out now. All right, get ready for this. Phosphates are the layers of membrane. Now they found these membrane layers, and in between they found clay, which they call it. The phosphate was found in asteroid Bennu sample returned by the OSIRIS-REx mission. Before they even came back with it, I knew exactly what it was. Well, you will in a minute too. There's no mistaking it. It's a heart. And the asteroid sample was returned here. Now the phosphate surprise, let me show you what it looks like. And remember heart sarcomeres. All right, so don't forget, these are heart sarcomeres, and this is ripped. All right, so what happens is when the heart's supposed to contract, it can't contract because it can't pull because of this. That's a heart attack. Now, these are the blocks. They're supposed to be like this, with the layer on the top, a layer on the bottom, phosphates, phospholipids, and these are the sarcomeres in the middle. Now, here they are anatomically. All right, this is the same thing as the other thing. This is just a flat plate representation, but there is a membrane at the top, membrane at the bottom. Well, what did they find in the samples from Bennu? Now, this is a stunner. I just showed you the heart sarcomeres. You know those little blocky things with a layer at the top, layer at the bottom. Well, what is this right here? This layer around here is a phosphate layer. They call this surprising phosphate findings in NASA. Now, there's another, this is where the sarcomeres are. There's a layer of sarcomeres, there's a layer of membrane, another layer of sarcomeres, and that's how they are, one after another, just as I showed you in that heart sample. So they're taking a picture of this with a certain type of light, and they can see the phosphates. Now let's see, they have some more pictures of this whole thing. Let me see if I can find it. Hold on. All right, I know, shocker. This is a heart sarcomere chunks, blocks, and it's ripped and it's torn. But they have the layers above and below, which are the phosphate layers, and you have the muscle in between. Here is what they found on Bennu. Exact same thing. All right, there's the phosphate layer. All right, this is when I see it with a certain light, and that's what it looks like without the light. But now they're going to show the layer on top and the layer on the bottom. So you got the layer on the top, covers the sarcomeres, and then you got the layer on the bottom. Now we're going to go in deeper and just look at this little spot, and here it is right here. There they are. Those are the muscle sarcomeres. They're laying on a membrane, and then there was a membrane on top. There's no difference whatsoever. It cannot be missed. Not only that, it has the phosphate surprise, which was the phosphorus, which is what the membranes are made out of. And this is nothing more than the clay, basically. And they admit all of this. They admit all of this stuff. Now, that brings us to my other research is about the fascia and about all these membrane layers. So there's a lot to learn from this. But the first thing is, you can see right now, that's a heart. That's Bennu. They landed on there, and he took a little chunk out of here, a little sample. Now, what happens with a heart? The tubing is glued in. It's literally glued to the valve set. This is the valve set. This is the plumbing. All right, so all this is muscles. And it's glued, literally glued. So this thing is doing this all day long. You can get smashed and bashed, and football players and car crashes and all kinds of things. And that will not rip normally away from the heart plumbing. It's a glue. So it can move around, and this is more stationary. But anyway, the heart is doing this all because it's really got to make a lot of action. And when you see really running and jumping and doing all kind of things, that baby takes off. You can pump 6 gallons of blood in 1 minute through your body with that little bugger. Now, what is this? That's Bennu. And what happens is when they dry out or when they get cooked or whatever, that just falls off. And you end up with the tendon, which is locked into the body, which is this tendon right here, and the valves underneath, and then some plumbing sticking out the side, and I'll show you that. This is it right here, and this is psyche, same thing. It's another heart, only this one smelted. It got next to too close to the sun or something, and cooked it, and it's metal on the outside, which is what blood is made out of, is metal, and this would do the same thing. This would do the same thing. You see the plumbing sticking out of here? Those aren't impacts, that's plumbing. And there's so much blood in the muscle tissues, it explodes when it gets cooked. And that's what most of these little things are, is popped out from being cooked. They're not just thousands of little hits on there. No. That's not it. And the plumbing breaks off. All right, now we're going to go into the spur lock. All right, now, I named it because I discovered it. I really started with the fascia long ago because I found it in the mud fossils and nobody could explain it. They thought there was a whole, they never even paid attention to it. Let's just go with that. And I said, no, this stuff is pretty important, and it seems to be one giant system, covers the whole body, and it's open, and there's fluid that can flow through here. And these membranes are really, really tough, and where they latch, that's the spur lock right there. Now, it's hard to see this one, but they come up and they service all of that membrane, which is the fluid-filled highway. That's what I originally discovered, was this giant fluid-filled network, is what I called it. I wrote a paper out in 2015. Now, but they all come down to here, and I never really thought too much about it, but one day I was looking at this rock here, looking at the spur lock, and I realized there's 2 little dots down the bottom, which are, and I mean they're small, but fluid from the lymph system, which this is the lymph, is the smallest fluids in your body. And there's 4 times more of this lymph fluid than there is of blood. It's all over the place. And it carries all the important things in your body, which is the enzymes, the bacteria, the littlest things that transport to do the feeding and oxygenation, all that stuff. not necessarily oxygenation. Your blood flows through a different system. This is extremely important, and I can demonstrate this in many, many, many ways. But let's look at this spur lock right here and what the actual plumbing looks like. Because I can show you. This is just, it's pretty incredible. When I saw it, I was shocked. All right, this is going to be cool. This is the rock that I'm going to be showing you in the microscope, which is this. And that is the spur lock, which is this in a living creature. Now, this is ripped, which is not supposed to be. Now, the fluids from this, whatever this was, I don't know what it was, but it has the same fascia coating, and in that fascia coating, there's a ton of fluid. It's a thin layer, but it's a bilipid layer. So there's a layer, then the lipids, and then another layer, and the lipids are where everything goes through. Now, this is what ripped off of here, and I have a lot of these that show the same thing. This is a tiny little lung, and that right there is the spur lock. And we'll look at that in a microscope. I got to get it wet, and then we'll look at it. The lung was here, and the plumbing went into the side of the lung right here. What it's from, I don't know. All the creatures died at the same time. I don't care what it was. And all of the pleura is gone. This is actually alveoli. When I put water on here, it'll take a whole different look. But that's the spur lock. They all have the same thing. Now, and I have them bigger and smaller and everywhere. And that's how things latch together in the membrane world. Even like this, look at this. This is the head of a bone. All right, so that's a bone. Now there's a spot you can see if you really look. Yeah, okay, right there. You see that coming up here, coming up here? This is the membrane. Can you see that? It looks like almost like a crescent roll. And then it locks in right up there. It's wrapped right around and it locks in. You should see some of them. It's just incredible. Some of them, they come like 4 or 5 places. And even like the no-toes, it's incredible. There's a latch. There's some kind of a latch that locks them in. And that's where that latch is on this one. Here's a triangle coming in like this, and there's the latch right there. And I call that the spur lock. They all have it. Every single one of them. So let's look at this one here in a microscope, and I'll show you these 2 little spots. And that, I'm almost certain, is where the fluids that were in there transfer into the next body part. Let me just show you something right here. Hold on, it's got to be here somewhere. Hold on, I got to go find it. Okay, this should make it as clear as you can possibly be. This is the spur lock. And coming out of there somewhere, I'm going to see if I can find it in a microscope. There should be 2 spots. That's the latch. This bag is completely sealed from the rest of the body. That's a lung. And it's been DNA tested. It's a human lung. There's no question about it whatsoever. And it died in a great flood, flat as a pancake. And I have many, many lungs. That's one that most of the pleura, well, not most, but a lot of it eroded off. And you see that layer there? And the rest is the lung. And that has the spur lock on the bottom as well. They all have it. Every single one. Because they have to transfer and the fluids from here, which is a special type of fluid as far as I'm concerned, and that's probably why there's so many cancers, because virtually every cancer is invasion. What is invasion? It's getting through the membrane and attacking the good stuff. So, and I've shown you can get blood out of a rock. You just drill little holes in them, get down deep enough, scratch out some dried stuff, take it out, and they'll put it in elutions and so forth, and they find it. Now, but this is what it should be, a rubber bag like this. All right, but they all died flat. Now, let's look and see what that looks like. All right, don't forget, we're going to be looking at that up above in a microscope, and it is that latch. Now, there it is in the microscope. I'm going to be jiggling it around here and there. You can see the triangle looking piece right here is the latch. Now I'm going to show you there's two spots there that it's transfer of the fluids. This whole thing would have been wrapped in a bag. It's broken apart. That's why you can see the blood so easily. Now I'm going to come down closer. We had to stand way off, but well, actually, let me jiggle it around and look. You see right back here? Let me see if I can move this. It's a little tricky, but I have a little clamp here that works very, very well. Now, that right there, that whole area there is like a little circular implant, and it is tough. And then you can see actually straps coming across. I'm going to come down a little closer, and you should be able, oops, you should be able to see a little better how strappy this is. Because this holds that organ in place. In this case, it's holding like a muscle to something. You see how strappy that is? And if we go back, it's just like that chicken. It's really, really tough in this one little gnarly spot. I mean tough, tough, tough. You see, these are all little straps coming together. And if you go back from that, it gets thinner and thinner, and then you get on top of the tissue, and it gets real thin, and it just turns into a regular membrane. But in this early attachment area here, it is tough. It's got to be, otherwise you rip you right out of your body. You just think of the stresses people put on their body. It's just incredible. You see some of these athletes, just nightmarish. In my mind, I can't imagine it. But anyway, this is the tough stuff. Now, where is the spots? They're right there, these two little spots. I'm going to come down real close. You see those two spots? That's where the fluid transfers from this bag into the next bag. Is this bag's fluid exactly the same as the next bag's fluid? I don't know. Nobody does. And that I can guarantee you. All right, what I intend to show in this little demonstration here is that there are two dots right here. And this is the triangular spot. I'm going to back it up so you can see better. As it goes back here, you see this is all very, very tough. And it gets back to this area back here where it is very, very tough. And that's the center of the implant. And going back from there again, it just keeps going back and turning back into fascia. It's hard to show it this way, but this is working pretty good. Now, coming this direction, we come all the way down to the front. And of course, this is just nothing more than really raw red meat. And that is the attachment spot. And those little dots, hold on, I gotta focus it back in, hold on. All right, we're going to play a little bit with the light just for kicks. This is real dim light. These are the two spots I'm talking about. This is just nothing more than flesh. And this latched this chunk of flesh into the next body part or wherever it was attached. Now I'm coming up with the light a little bit. A little too much. And you can see these are very, very, very tough things, but this is where the fluid is passing. And again, my question is this fluid exactly like where it's going into the next organ? And what would happen if one of these holes got plugged up and this couldn't get fluid in it? There's 200 types of cancers, and every one of them basically relates to one of your body parts, and every body part, I'm almost absolutely certain, has a different chemistry surrounding in that membrane. I would love to see autopsies done and doing the chemical composition and molecular composition of what's in these membranes. What kind of bacteria live in there? What kind of organisms live in that layer? Because that layer is the most important layer in the body. That's the membrane, keeps everything out and keeps everything in. It only allows the right stuff in and pushes the bad stuff out. That requires a lot of chemistry, a lot of chemistry. Okay, as you have seen or will see, I have more evidence than the average researcher on this, and I feel that I have come to some valid conclusions. And what does all this mean? All of your organs are in their own specific chemistry in a bag, I believe. Then that chemistry leaves through this to another organ, which will have its own specific chemistry, I believe. Totally unknown at this moment. I think from autopsies we could get an idea if that's true. But we certainly can test it in one way or another. Secondly, could these plug up? And what would be the consequence if they did? And there's more to this. How does acupuncture work? There's 400 or more acupuncture sites on the human body to do all kinds of things. Now, do they work? Nobody really knows. But I looked into it because I'm thinking maybe this is going to be sites that are acupuncture sites, and it does appear that some match up, but there's 400 of them. You got to hit one here and there. Now, what is the effect of acupuncture? What does that do? It puts a hole in something. Well, why is that good? What is the release of your body when you get a hole in it? It creates inflammation to some degree. Now, these are very, very fine, smooth, really fine needles, and they can basically wiggle their way through most of the tissue. So there's not a whole lot of damage. They're not tearing and shredding things like a knife would do. However, they have to create some kind of a response. There's just no other way. You're being invaded by something. It's not going to say, okay, that's fine, have a nice day. Although it might, I don't know. At this point, I'm sort of up in the air about a lot of things. But there's a lot of things that haven't been examined. That's why I'm up in the air about them. Nobody knows. Nobody knows about that. Nobody knows about the Spurlock. I've checked with a whole batch of different anatomists that are very, well, not a whole batch, but enough that I realize they have no idea what this is. And again, the interstitium layer, which I wrote the paper on about 2015, was just recognized in 2018. And they're still deciding, is it one giant organ? And yes, it is. However, these different cells of this organ are capsules that incorporate all the different types of body tissues. So if you don't have the right chemistry inside that cell that surrounds whatever that organ is in peril. Okay, so my buddy Chat and I were talking, and I said, well, how does it work? How does acupuncture work? And Chat said, acupuncture believed to work through a combination of mechanisms, though its exact process still a subject of research. Here are some key theories on how acupuncture may be effective. Meridian theory, I don't know about that. That's way out of my wheelhouse. Neurotransmitter release, yes. That's something in a neurotransmitter release. Some studies just acupuncture stimulate the release of something that's trying to affect, like in other words, it says, go over there and see what's going on. There's somebody trying to invade. So these neurotransmitters go over there and they say, hey, what's going on? What was that? The guy just stuck me with a pin. And then they send over endorphins or serotonins or enzymes or whatever to not be damaged. And they help reduce the pain and improve the mood and so forth. They don't really know how it works. I don't know how it works either. But the blood flow, maybe. Inflammation, yes. There's going to be some of that. Acupuncture maybe increase blood circulation, can aid in healing, reduce inflammation, nervous system effects. At this point, nobody knows. But I would like to know about this thing up here and about those fluids, because that's the key as far as I'm concerned. Those fluids that are going back and forth through the different organs are the key to your health. They contain the fluids that do everything in your body. They digest your food, they transport your minerals and tiny little enzymes and chemistry and bacteria, and they lay in that layer that says, don't you come through here, it will kill you. That's your membrane.